Bismillahirrahmanirrahim. We now continue with the previous paper that I started, and this will be the second video. And I will try to cover questions number 4, 5, 6, and if you have time, we can do 7 and 8. So, March 2019, paper 4-2. The main art of doing a paper 4 is to read the question very carefully. So, Mexican spade-foot toads, well, whatever the name, live on land but return to ponds to breed. Eggs are laid in water and hatch into tadpoles which feed in ponds before developing into adults. The tadpoles can be classified into two main types, omnivore type tadpoles and carnivore type. So we only have two types of tadpoles, the omnivore type and the carnivore type. Differences between the phenotypes of these two, phenotype is physical features, are related to their different feeding behaviors. So the first information they give you is that omnivore type tadpoles feed on tiny pieces of detritus, dead material from plants and animals, and algae, which are unicellular plants. At the bottom of ponds, these tadpoles grow very slowly. Okay, so we've got very important information is omnivore type grow slowly. Then the carnivore type. Now, if you look at the carnivore type tadpoles, we have now another type, feed on small animals in the water, so no plants, such as fairy shrimps. And small carnivore tadpoles, these tadpoles, they feed on shrimps and small omnivore tadpoles. And these tadpoles grow very quickly. Okay, so we have two pieces of information. The two pieces of information are omnivores grow slowly and the carnivores grow quickly. So two pieces of information. Now you can see how I have just outlined or just marked them. Omnivore grow slowly, carnivore grow quickly. Now figure 4.1 shows two tadpoles of the same age, one of each type. A fairy shrimp is also shown. All these organisms are all at the same distance from the camera. Okay, so we have this picture which you can see there is the carnivore type tadpole, which is of course definitely much bigger, and then the omnivore, which is smaller, and then the shrimp is the smallest. Now you know the carnivore eats this and this, while the others only eat the dead matter of plants and animals. Now, between these two main types of tadpoles, now let's look at the information given to us. Between these two main types of tadpoles, there is a continuous range of tadpoles with intermediate body phenotypes. So some shorter, medium, all different lengths and different feeding behaviors. Some eat only plants, some eat both, some only eat the shrimps. So for any individual tadpole, regardless of age, it is possible to calculate a phenotype score depending on the features of the tadpole. A tadpole with a phenotype score close to 3 is a typical omnivore, and a tadpole with a phenotype score of 7 is a typical carnivore. So 3 typical omnivore, 7 typical carnivore. The phenotype scores were determined for a large number of tadpoles sampled from 2 ponds. The availability of detritus is dead organic matter and algae is the unicellular plant was high for one pond and low for the other pond. All other conditions in the two ponds were similar. For both ponds, phenotype scores were determined shortly after the tadpoles had hatched from the eggs and 10 days later. So read the question very carefully, underline the keywords, and explain this question to yourself. First of all, you must understand the question and then see how you can handle this. And then you should read the whole question before you start answering it. Then you see, then it goes on the question. The results are shown in figure 4.2. And you can see a phenotype score here, which is on the x-axis, and you see a phenotype score here. Here you see there is a high availability, and here there is a low availability. But the frequency is nearly the same. The three are found in smaller numbers, seven, and in between are found in large numbers. Similarly is the case here. It's nearly the same case here and then. So look at the graph, see what is the phenotype score, 3 and 7. 3 are less in number, 7, but the middle ones, maybe 5, are more in number, frequency. And then, of course, 10 days later, what do we see? Now, 10 days later, the graph seems to change a bit. In this one, the phenotype score 10 days later, in the one which had high availability, it stays the same. So high availability of detritus remains the same, but in the low availability of detritus and algae, 10 days later we see it changes. You see the 3 are now more abundant and the 6 are now nearly more abundant. And the ones in between, the 5 ones are somehow at a disadvantage because you see them, they have decreased. So this one has now decreased. So the frequency has decreased while here the frequency was here. So you look at the graph, you explain the graph to yourself, you do all this, what I'm doing on the graph, making these lines and explaining this to you all. So explain this to yourself. I know you don't have a lot of time, but you have a mark, a minute for every mark that you need to score. So you need to spend this time and understand this question before you can really proceed. Now, A part says, using the results shown in figure 4.2, describe and suggest an explanation for the change in frequency of tadpole phenotypes in the pond with low availability. So low availability was only asked to be discussed, not the high. So frequency of extreme phenotypes remain high, right? Disruptive selection, this is called. Selection pressure is food availability. So extreme phenotypes have a selective advantage and they are good at accessing the available food, and there is competition for food. So in that you could see why the extremes of phenotype were at an advantage, so their frequency increased. So extreme phenotypes have a selective advantage. This is the English that we expect you to write when you're doing these questions. So why were they at an advantage? Is because they could eat more of the food, whatever was available. So whatever food was available was the ones who were good at accessing that food or digesting that food or eating that food or finding that food, they were the one who were the winners. And they, of course, increased in numbers, and their numbers or their population increased because they were at a selective advantage. Now, these are the words that we use: selective advantage. Extreme phenotypes have a selective advantage, and this is called disruptive selection when you have two peaks. Then part B says 11 days after the eggs hatched, the mean body masses of omnivore tadpoles and carnivore tadpoles in the pond and low availability of detritus algae were measured. The results are shown. We just have to read off. So y-axis, using the data in figure 4.3, calculate the mean body mass of an omnivore as a percentage of the mean body mass of carnivore. So show your working, and that was the answer which you had to give me. The C part of the question was the phenotype scores of a large number of tadpoles were determined in a different pond with very low availability of detritus and algae. All other conditions were the same as the first two ponds. As previously, measurements were taken shortly after the eggs hatched and 10 days later. After 10 days, nearly all of the Mexican spadefoot tadpoles in the pond were carnivorous types. The results are shown in figure 4.4. Pond with very low availability of detritus and algae, frequency 10 days later. Now the phenotype score, you see how it has changed. State the type of natural selection that is acting on the tadpoles in the pond with very low availability of detritus and algae. This is called directional selection when the mean changes. So this is called directional selection. We all know that, you have to just name it. Then it says suggest explanation for the change in phenotype frequencies of the tadpole in the pond with very low availability. Omnivore type die. You see, 3 was the omnivore type and 7 was the carnivore type. So omnivore type die, the carnivore type survive. Selection pressure acts against the omnivore. The omnivores were somehow at a selective disadvantage. So they all died, and now you can see that the 7 ones are living. So three were the ones which were omnivore. So omnivore type die, carnivore survive, selection pressure acts. Now this is the English that we expect you to be writing is selection pressure acts against the omnivore type. You could have said the phenotype score 3 or the phenotype score 7. That was also allowed. Last part of this question was simple. Mexican whatever toad tadpoles develop into adult toads that do not live in water. In some years, the ponds where Mexican spadefoot toad tadpoles live dry out quickly. Suggest why the carnivore type tadpoles have a selective advantage in the years when ponds dry out quickly. They grow quicker, so develop into adults in shorter time, or they develop into adults faster. So they would not be bothered about staying in the pond, and they could reach, and then they could. So it didn't bother them if the ponds would dry out, and they would be able to survive. Look at question 5. Question 5 is about now GM crops. So figure 5.1 shows the area of land that was used to grow genetically modified GM crops in the USA, Brazil, India, and China from 2004 to 15. So this is for USA, this is for Brazil, this is for India, and this is for China. So area of land used to grow GM crops in million hectares, and this is the year. So you look at the graph and you see how the area of land used to grow GM crops in the USA is increasing. Now, what you need to do is you need to talk to yourself and explain this to yourself is that USA, you see how the USA is really increasing. I've given it a green line and you can see how it was here, here, and then now it is here. So it has really increased the area of land used. Then Brazil has increased, but it has increased less than the USA. Then India is the one which I've colored in blue, and you can see India is only gone up a bit. It's gone up to 10 million here, so nearly 10 million here, so we write 10 here. And then China has remained constant. China has remained constant. It has remained constant. The line has remained the same all throughout the 2004 to 2015, which means 11 years. So you do this whole story to yourself, and you, of course, I'm not saying you use colors, but you do this to explain this to yourself. Then in part A1, this has suggest reasons why the area of land used to grow GM crops in the USA is greater than the area of land used to grow GM crops in Brazil. Now, they don't expect you to know a bit of geography, I guess. So can afford GM crops developed in USA, more land available to grow crops, more public approval, you see, because in certain countries like Europe, the public doesn't allow you to grow GM crops, and they're very, very staunch and they're very clear-headed. They do not want any GM crops. So more public approval or the climate is suitable for GM crops. So any two of these, and you got your 2 out of 2. So suggest is the one which is always very, very difficult, and it just requires a basic knowledge and understanding of biology, and there's no one answer to it. Part two of the question was describe the differences in the area or land that was used to grow GM crops in China and the area of land that was used to grow GM crops in India. So you are expected to do a comparison between China and they used to grow GM crops in India over time, shown in figure 4.1. And I told you describe is no biology, so you have to just give me a comparison of what was happening. Now, if you look at the graph, you can see the comparison between India and China. So India and China here, I've labeled these. Now, initially in 2004 to 2006, if you look at it here, India uses smaller area than China. So 2004 to 2006, India uses smaller area than China. So you can see the blue line was below that. Then area in China remains almost constant. So China has remained almost constant in this whole story. Then areas in India increase throughout. You can see here areas in India, which is the blue one, increases throughout. There's a slight increase, it keeps on increasing and it keeps on increasing. Then area in India is greater than China after 2006. So area in India is greater than China after 2006. So you realize whenever there's a cross, when one line crosses the other, that's the point which you need to be making. That's the point they expect you to come out with. And then if you give me comparative figures, one area from India and one from China, one area from India and one from China, if you gave me that, and for 2 different years, say you gave me in 2010 and then you gave me in 2015, and you gave me the area from India and one from China, and for two different years and plus the units. So that would have got you another mark, and that's called a data quote or a comparative figures, and that could have got you the mark for that. You can just read the mark scheme in 2004, 2006. India uses smaller area in China constant, area in India increases, area in India is greater after 2006, and then if you gave me a data quote or comparative figures, you got another mark. But there were only 3 marks for this, so any 3 out of these would get you your 3 out of 3. Then for part B1, it was a simple genetic modification in crops can provide resistance to insect pests. Bt maize is one type of GM crop that has been produced to be resistant to insect pests. State 2, benefits to farmers of insect resistant crops. Naturally, when the insects are not going to eat their leaves and they are not going to damage the leaves, so more photosynthetic area, more glucose, and more food, so increases the yield of the crop, increases the quality of the crop. No insecticides need to be used and no money spent on insecticides. Then it says describe the difference between Bt maize and non-GM maize, that explains why Bt maize is resistant to insects. You see, what have we done when we've genetically modified it? We've taken a gene from a bacteria and we've put it into the maize crop, and that produces a Bt toxin which is harmful to insects. So the gene we've taken from a bacteria, then we put it into the maize crop, and now when the caterpillars come and eat the leaves of that, they die. So it is toxic for the insects, so it's harmful to the insects, and the insects die. Then it says C1 is state two reasons why people may have objections to the growth of insect resistant GM crops. In fact, I have a lot of objection. I'm very much against GM crops. So resistance to insect transfer to the wild plants. It may kill very useful insects, you saw, which are pollinators, and that was going to result in decrease in the biodiversity. And then we don't know. I mean, after many, many years, we'll suddenly come to a conclusion that after people who've had GM crops develop these diseases. So we have no idea. I mean, we haven't done any research on that. That does GM crops do any damage? Maybe the reason why people have more hypertension and diabetes and cancers will all go down to GM crops. So it's health risk. So people have a lot of issues about it. So growth is too why people may have objections, health risks to humans. And because no research has been done on this, and it's all a whole body of people who just advocate this to increase crop yield and make more money. Then we come to the part 2 of the question. In 2015, the percentage of the USA population that objected to the use of GM crops was lower than 2005. So, I mean, people, not that people are becoming more intelligent or more dumb, but it is why a smaller percentage of the USA population objected. Now, reasons are for objections was not proven. As far as I think, it is people just did not know what they were eating. So GM food consumption shows no ill effects. That's what the media says. That's what all the social media says. And entire generation grown up in the GM era, so they have no clue. They don't even know what was times when there were no GM crops. And of course, and the last point is education and awareness. I don't think, frankly speaking, I don't think the majority of people even know anything about GM crops, and it's a very small percentage who would be educated enough to really say that, okay, we're against GM crops. So, but I mean, for the paper's sake, people did not know. They felt that the objections were not relevant or that the food consumption did not show any ill effects. People were not dying of it. So people were eating it, and the entire generation had been eating GM crops. So they really didn't know what they were doing. So this is the answer to that 1 mark that you needed to give. Now let's look at question 6. Mila syndrome is an inherited disease caused by a mutation in a gene located in the mitochondrial DNA, empty DNA. And all empty DNA is inherited from the mother. We've studied that before, is that, because only the nucleus of the sperm fuses with the ovum. So all the cytoplasm from the ovum, which is the mother's cytoplasm, will go into the zygote, and the zygote will contain mitochondria, which is from the mother, and that is called mitochondrial DNA. Now, figure 6.1 shows four generations of a family where several individuals are affected by this Mila syndrome. And the first generation we had 1 and 2, but then if you look at 1 and 2, 4 and 5 are the offspring, they both have the disease. And then fourth is the female, and then her son, daughters all have the disease. So 3 and 4 have three kids, 8, 9, and 10. Now 5 and 6 have three kids, but none of them have the disease. Then 10 has the disease, so female with Miller syndrome, and then 11 doesn't have it, but both the kids have it. So you see, in what they're going to see is that you're going to read this and you're going to understand this very carefully. So unaffected female, unaffected male, and then you have the female with Miller syndrome, and then you see how one doesn't have it, the one doesn't have it, two has it, but both the kids have it. Now, with reference to figure 6.1, state and explain the evidence that Miller syndrome is an empty DNA. So they want you to explain that it is an empty DNA disease and not a disease caused by a mutation in a gene on the X chromosome. Now you have to understand is if this is a sex-linked disorder, then more males would be affected. But here this is not the case. More females are in fact affected. So explain the evidence that this is empty DNA disease and not a disease caused by mutation in the X chromosome. Now, if you look at the mark scheme is very categoric. Miller syndrome is not inherited from affected males. You see, in sex-linked disorders, it is the male who are more affected, and the disease is passed on from the affected male. All offsprings of a female with Miller syndrome also have Miller syndrome. Then offspring 4 and 5 of mother 2 have Miller syndrome. Then males more likely to be affected. If I have to talk about empty DNA, there were two headings you have to talk about. If you're proving empty DNA, if you're proving X-linked, then in the X-linked, males more likely to be affected. Males inherit only one X chromosome from the mother. More females affected than males. In this situation, in figure 5.1, more females are affected than the males. And there are no carrier females. So this was this possible answer you had to give me for 4 marks and 4 points. Any of the 4 points would have got you the 4 out of 4. The B part of the question says analysis of empty DNA can show how recently species have evolved from each other. Describe the properties of empty DNA that makes it suitable for the study of evolution. Now, the first thing is mutation occurs at a constant rate, faster rate than nuclear DNA, and the empty DNA is not protected by the histone proteins, and there are no enzymes to repair DNA mutations, and many copies of the empty DNA per cell. What does that mean? One cell has many mitochondria in it. In fact, some cells have up to 1,000 mitochondria. So many copies of the empty DNA per cell. That is the important word that you're supposed to use. And circular DNA. Why? Because mitochondria have a circular DNA. So there's no crossing over. Crossing over can only occur between linear DNA, chromosomes. So there's no crossing over here. And that, of course, is why it is a better idea to study these when we are studying the study of evolution. Do question 7a. Isolated mitochondria were used to investigate respiration. Mitochondria were extracted from respiring mammalian cells and incubated in a buffer solution. Pyruvate and inorganic phosphate were added at time 0. ADP was added 1 minute later. So here ADP was added 1 minute later. And pyruvate and inorganic phosphate were added here. So pyruvate and inorganic phosphate were added at time 0. ADP was added 1 minute later. The oxygen concentration of the buffer solution containing mitochondria was monitored throughout the investigation. The results of the investigation are shown in 7.1, and you can see in phase A, what is happening is in phase A, we have the oxygen concentration of the buffer solution containing mitochondria. So here we have on the y-axis is the oxygen concentration, and you can see the oxygen concentration is slightly decreasing in A, and it's very rapidly decreasing in the B part. So oxygen is being used up. And then it's a slight, in phase C, of course, we have the oxygen decreasing less sharply. So you've got all this, you've got to explain this to yourself. Pyruvate and inorganic phosphate added here at 0, and then ADP added here, and as soon as we add the ADP, then the oxygen level falls very, very rapidly. Part one of the question suggests why the line of the graph on figure 7.1 is steeper during phase B than during phase A. Yes, because faster rate of aerobic respiration. That's why the oxygen is being used up. So the oxygen concentration is falling very rapidly. In phase B, ADP is the present. So oxygen concentration decreases faster. You see, more ADP means more ATP. Oxygen is the final electron acceptor. So ADP needed for ATP synthesis and oxygen used as a final electron acceptor. And you had to give me any 3 points for this. Suggest why the line of the graph starts to level out in phase C. Naturally, because something is running out. And it could be ADP, it could be PI, the inorganic phosphate, it could be oxygen, it could be pyruvate, and that becomes the limiting factor. So all the factors, whenever something levels out, there has to be something which is limiting it, and that maybe something is really decreasing the rate. That's why we do limiting factors in photosynthesis. Come to the B part of the question, and the B part of the question says some plants such as rice grow with their roots submerged in water. Figure 7.2 shows a group of rice plants. Explain how rice is adapted to grow with its roots submerged in water. It was a very direct question, and if you knew it, well, you knew it. Otherwise, you just could be making this all up. Low concentration of oxygen in the water, aerenchyma is present, these air spaces in the leaf and the stem, and the gases diffuse down from the leaf and down to the root cells, which does not happen in other plants. This allows aerobic respiration. So because the oxygen is going to diffuse down to the root cells because there's too much of the roots are submerged in water. So how are they going to get oxygen? Usually the roots, the land plants have air spaces in the soil and that provides it the oxygen. So we have some leaves trap air underwater due to ridges on the leaves. Ethanol produced from respiration under anaerobic conditions. Root cells can tolerate ethanol. So the fact that root cells can tolerate ethanol is of course an advantage, and that is because good for them. And the reason is the root cells produce ethanol dehydrogenase, which of course breaks down the ethanol. And then some rice varieties have a high rate of respiration even in anaerobic conditions, and it can still generate more ATP. So these are all the factors which help the rice to be able to survive these anaerobic conditions. And that finishes this video, and we'll continue with the other questions left in another video. Thank you very much.